Hi everyone, my name is Lot Kingori and I'd like to thank you for enrolling this masterclass of Formulation Development, a risk-based approach. Just to give you a bit of a background of my academic qualification and professional experience, I have a Bachelor of Pharmacy degree from the University of Dar es Salaam and a Master of Science in Pharmacy majoring in Pharmaceutics from Rhodes University. My research thesis was focused as using enhanced quality by design in developing and optimizing formulation of a fixed dose combination of ranitidine and metronidazole. Yes, I know it's a bit of a mouthful. I have a 20-year work experience in the pharmaceutical industry in the areas of production, pharmaceutical development, and manufacturing process efficiency. So in the next 2 hours or so, I'm going to share my knowledge in formulation and manufacturing process development that I acquired throughout the course of my career. I hope you get to enjoy the course. So I'm going to share my screen so we can begin. We're going to discuss about, it's like how the lectures, the course, it's described, formulation development, a risk-based approach. So I'm going to just give you a brief overview. So we're going to talk about quality risk management, quality by design, and the application of the quality risk management, quality by design in the developing of formulations, which is a risk-based pharmaceutical development and pharmaceutical development reports. And yeah, and knowledge assessment discussion at the end. So basically just a short quiz to test your knowledge in what has been discussed. So pharmaceutical development. What do we mean by pharmaceutical development? So quality by design, which is commonly used in the pharmaceutical development or in formulation development, is a designing of a quality product and the respective manufacturing process that consistently deliver the intended product performance. So you can see that I have highlighted quality, the word quality. So what is a quality product? A quality product is the one that is safe and efficacious. So safe in the sense that it has tolerable side effects, because there is no product which doesn't have a side effects. Some are severe and some are less severe. So it should have a tolerable side effects and efficacious, which means that it should be able to elicit a pharmacological response and also to meet users' expectations. So how do you design a quality product? You do it by building quality into a product, by applying scientific approaches and quality risk management to development of the product and respective manufacturing process. So what do you mean by scientific approaches? So basically, it's an understanding of unique chemical and physical properties of the material and the components that are used in the manufacturing the product. Because you need to have a thorough knowledge of the ingredients that you're using, because this would enable you to do a risk assessment. Because at the end of the day, you have to incorporate risk. What are the possible effect that could happen to a material that could stop it from doing what it's supposed to do, to behave, to act like what it's supposed to do. So you want to eliminate those risks. And the only way to be able to eliminate the risk is to have a thorough understanding of the chemical and the physical chemical and physical properties of the materials. So that's what we mean by scientific approaches and quality risk measurement. So the thorough understanding of the material, the chemical and physical properties of the material, coupled together with risk analysis to be able to ensure that your ingredients and the process, they are all do not antagonize each other to enable for you to develop a quality product. So QBD is the recommended approach of pharmaceutical development by most regulatory authorities, because basically it employs risks analysis together with scientific approaches. So what is quality risk management? So it's a systematic process of assessment, control, communication, and review of risk to quality of a drug product across the drug product lifecycle. So it can be applied both proactively, that's before a failure occurs, or it can be retrospectively after a failure has occurred. The evaluation of the risk to quality is based on experience with the process, usually, but not always, undertaken, and usually, but not always undertaken by a multidisciplinary team. So you need to have a thorough knowledge of the process or to be able to do a risk assessment. Because without having a thorough knowledge, you won't be able to predict, I mean, to understand what could be the possible failures that could happen to cause that harm or deficiency in product quality. And the effort and formality and documentation of the quality risk management process should be commensurate with the level of risk. So what do we mean over here? So the amounts, the extent and the detail that goes into the risk analysis and the formality of the process and the level of documentation should be should be similar to the type of risk. That means if it's a small risk, like for instance, you want to change a shipper for packing your product, a shipper which is the box. You want to change from one supplier A to another supplier B. You don't need to put too much effort to understand what could be the possible failure could happen, what are the risks to product quality, because it's just a shipper. This is a packaging, it's a box. It's not even a packaging that is in direct contact with your product. It's just a shipper, which is the brown box that you use for shipping. So you shouldn't spend too much time trying to understand what could be the possible risks of product quality when you're changing shipper. But if say you're changing a primary packaging material, which is your say your bottle, which is in direct contact with your product, then you need to put too much effort and time and formality because the risk to product quality by changing primary packaging material from supplier A to supplier B could have could be of serious consequence to your product because this is a material that is in direct contact with your product. So you need to put more time and formality and the level of documentation because the risk is high. So that's basically what it means. So why do we do quality risk management? It's an effective quality risk management system can result in improved decision making in the event of a quality problem arising. Let's say if we've noticed that there is there is stability failure. So what do you do? Do you pull the product that's already in the market because there's stability failure when you're doing the testing, or you can do you leave it in the market and say, okay, because there's instability failure, the failure is unlikely to have an impact on the quality of the product. So that's where the quality risk management comes in. You need to analyze the risk and say, okay, based on the risk assessment we have done, we see that the impact to product quality is minimal because the related substances are still within limit, but the change is only in color, which does not have an impact on the efficacy and the safety of the product. So we've decided to leave the product market, or we can say the impurities are relatively high, so which can pose a risk to the patient. So we decided to recall the product from the market. So that's one of the why you should do a quality risk, why we study quality risk management. And secondly is to ensure product quality is maintained throughout the product lifecycle, which means attributes that are important to the quality of the product remain consistent in case of a change. So what do we mean by this? So if you want to change, let's say, raw material, which happens very often in the pharmaceutical manufacturing, we want to move from supplier A to supplier B. So before we make this change, make it formal, we need to do a risk assessment to see whether there is similarity between the material from supplier A and supplier B. Because if there's no similarity, then that means if we proceed with the change, we might have an impact on the change might have any change of using supplier B might have an impact on product quality. So you need to do a risk assessment before you decide to implement that change. So risk assessments can also be integrated into existing operation and documented appropriately. So you should have a defined approach of a risk assessment. It should have an SOP for it, how you're going to approach it, such that whenever that you need to implement it, we know there's this SOP and it can be implemented seamlessly. And the reason that we want to do that because change is inevitable in the pharmaceutical manufacturing, where there's material, or you can have deviations, or you can have incidents that happen, and you need to apply risk assessment in order to be able to make correct decisions. So what is risk? Risk is a combination of the probability of occurrence and severity of following exposure to hazard. So that's what we call it risk. So there has to be probability of occurrence and severity of following exposure to hazard. And what is hazard? So hazard is a potential source of harm. So how do you identify hazard in the risk assessment? You need to start by a well-defined risk question or a problem description. So in order to identify a risk question, you need to have the following guidelines, and which are the three fundamental questions that aid in identifying the problem description. And this is, you might ask yourself, what might go wrong? And secondly, what is the likelihood, which is the probability of it going wrong? And thirdly, what are the consequences or severity of whatever that's going to go wrong? So these are the three fundamental questions that you need to ask yourself in defining a risk question. So example of a hazard identification. So for instance, the problem description or risk question is, we're changing raw material from supplier A to supplier B. So what could be the risk over here? So we start by defining the risk question, which is the dissolution rate of the active ingredient might be out of specification. That is what might go wrong. And what is the likelihood of probability of it going wrong? It is likely. And what is the severity? The severity is the efficacy of the drug product will be compromised. So these are the three fundamental questions that we ask ourselves in the defining the problem description or the risk question. Risk question. So what are the enabling factors in identification of hazards? So you need to use a systematic, you need to have a systematic use of information, such as you need to have a prior knowledge of that particular process or of that particular uh, experience, or you can use historical data, which is if you have been, say, manufacturing a particular product for a period of time, you'll have a certain data regarding that product. And now that we'll be saying that we want to switch raw material from supplier A to supplier B, you can see that, okay, whenever that we've changed raw material from supplier A to supplier B, we see dissolution pattern to change. Maybe where there's amount released in 45 minutes, it's changed from 95, now we're seeing 88% whenever you supply B, but when you go to supply A, it goes back to 95%. So you have that data so that you can leverage that in identification of hazard. Also, you can have a theoretical analysis, like scientific knowledge on material and physical chemical properties. Like for instance, like theoretical analysis is, like say a change in particle size. Say supplier A has particle size of average of 90 micrometers and supplier B has a particle size of average, say, 50 micrometers. So the lower the particle size, the higher the surface area. So you expect maybe dissolution rate to be higher. And the bigger the particle size, the lower the particle size, the higher the surface area. The bigger the particle size, the lower the surface area. And if the lower surface area, we expect that the dissolution to be impacted to go low. So that's your theoretical analysis. And also another thing you can use is expert opinion concerns. This it's used in cases where there is no empirical data or information available on the variable of interest. So you don't have, there's no information to justify that. So basically, you can just say, okay, we expect because of a change in raw material, we don't have data on the supplier, we don't have information. There could be a risk that maybe our dissolution might drop, might be impacted. So it's worth doing studies on this before we implement the change. So that's how you identify hazards you need to have, as you said, you need to you need to have a systematic use of information for you to be able to identify hazards and consequently the harm associated with those. So risk analysis. So risk analysis is a systematic, qualitative or quantitative process of estimating risk by linking the likelihood of the occurrence and the severity of the harm associated with identified hazard. In some risk management tools require the inclusion of detectability of the harm in the estimation of risk. So, but in pharmaceutical processes, we often include detectability in the process of doing a risk analysis. So we use the likelihood, which is the probability, and the consequence, which is the severity, and detectability. All of these three are linked together in order to estimate the harm associated with exposure to that hazard, which is a risk. So pharmaceutical processes and procedure require the inclusion of detectability of the harm in the risk analysis. Risk analysis can be undertaken qualitatively or quantitatively. Qualitatively usually employs classifying the risk, whether it's high, low, and medium, and quantitatively usually employs having a number which can weigh the gravity of the risk. So the higher the number, it implies that the risk is, or the harm, or the failure is more, it is high risk, which is of high risk or of high consequence. And the lower the number, which means the risk is of low consequences. So in this way, you'll be able to gauge and decide your control measure, how to prioritize and the extent of the control measure that's required depending on the number that you've attained in the risk analysis. So the outcome of a risk analysis is a comprehensive list of risk events, mapping out the probability for the risk, severity, and the detectability. And this will decide how you respond, how you design your control measure. So qualitative risk analysis requires a determination of risk class, which is your severity times probability of the harm or failure. And thereafter, you define the risk priority, which is a multiple of your risk class times the detectability to classify the risk. So qualitatively, like I've said before, you don't allocate the weight, which is your number. And in this case, you just classify whether the risk is high, medium, or low. And quantitatively, you do allocate the number, which can help you to quantify the risk. And based on the number, you should be able to know whether it's high. You can say, okay, this is what I need to do to allocate more attention to the high risk, which is a high number, and less attention to the low risk, which is a low number. So qualitative risk assessment, like I said before, you need to identify the risk class. So you have a severity, which could be high, medium, and low, and the likelihood of occurrence could be high, medium, low. And the product of that, which is your severity and the probability, you get the risk class. And the risk class is a multiple of your severity and the probability. Like for instance, if you look at the severity, if your severity of the is high and the probability of that occurrence of harm is high, then your risk class will be high, which is a multiple of high in severity and a multiple of high in probability, and you get a high risk class. If your severity is low and your probability of occurrence is low, then your risk class is defined as low. As you can see I don't know if you can see my pointer. Let me see again. Okay. You can see over here where it's a multiple of low from severity and low from probability to get your low risk class. And if you say the severity is medium and your probability is high, then your risk class is categorized as high. And there, once you identify the risk class, we can move to this matrix over here, where you have your high, medium, low risk class. which is these categories over here, just being called a bit uh, a line over here, which is high, medium, low. Now you include the detectability, because in the pharmaceutical process, you won't be able to detect a particular failure. So if you can see a failure, if you're able to detect a failure before it happens, then you minimize the failure. You can minimize the failure from occurring and the impact of that failure on the product quality. So you want to be able to include detectability in when you're doing your risk analysis. So if you have high risk class and a high detectability in risk class, it means that the possibility of you detecting that risk is not really high, but it's difficult to detect. And detectability is the other way around. So it's difficult to detect. That means that risk is classified as high risk. That means detection is almost impossible to detect the failure. So the risk is classified as high. But if detectability is low, that means it can easily be detected before the failure happens. Then you describe that detectability as low detectability, low detection. That means you don't need to use sophisticated instrument. Basically, you don't need to have instrument. You can just basically detect it visually. Then we say that is low detectability. So a combination of a high risk class and a low detectability, then you get a risk priority of medium. So if you have a low risk class and low detectability, then overall risk priority for that particular failure is classified as low. So once you have identified your risk priority, whether it's low, medium, or high, which is all the values in this matrix over here, which are listed over here, low, medium, and high, then you should be able to know what sort of action needs to be taken. So in case if your risk priority is low, then you say this is a low risk class. And in this case, this class can be accepted and improvements doesn't, we can decide whether what do we need to improve. And it's not necessarily has to be urgent because it's a low risk. And if it's medium, then requires additional control. You can say, okay, this is a medium risk. What do we need to do? Do we need to put a procedure in place? How are we going to handle this? Or do we need to do some more tests of the raw material before we implement the change? Or do we say, okay, we think the risk might give us a problem in the manufacturing processes, so probably we shouldn't proceed with this change from supplier A to supplier B. And if it's a high risk, then you can say, you know what, we're probably going to encounter a lot of batch failure by implementing this change. So we're just going to continue with our existing supplier. And if we're going to lose the market share because our product is still going to be, it's going to be expensive from those of the competitors, that's fine. We are happy with that risk. So that basically that's how you do your qualitative risk assessment. So now we're going to look at quantitative risk assessment. So quantitative process of estimating risk entails determination of the risk priority number, which is given by the product which is a multiple of the severity, occurrence, and detectability. So what is severity? So severity is the degrees of seriousness of the harm, the impact on the patient safety, product quality, and doctor integrity, or any other harm. So when you look at severity, you have to see, okay, if this failure happens, what is the impact of this failure, of the harm, or the failure on the product, on the patient safety, or the patient, or on the product quality, or on the data integrity, or any other harm that you can think of. And occurrence, we're referring to the likelihood of the fault or failure mode of occurrence. Is it highly probable or is it less probable? Out of 10 incidences, what is the likelihood it's going to happen? And detectability is the likelihood the fault will be detected. So if the fault is hard to detect, then you say that's high then you deem it as high. If the fault is easy to detect, then you say detectability is low. So the risk priority number, which is a numerical value, is used to prioritize the failure mode based on the severity of occurrence, severity and the consequences and the detection score. So basically, like I said, is a multiple of your severity, occurrence, and detectability of the failure. So that's what we use in quantitative risk analysis. So in other words, you can rank your severity in either, you can have a number of 1, 2, or 3. So if a severity you say has a value of 1, what does that mean? So this value of 1 means like possibly the consequence, there is no consequence with that particular harm. If that it happens, the severity, the consequences are negligible or there's no consequences. So negligible impact on patient safety, product quality, and data integrity. So the failure or the harm has no impact on the patient safety, product quality, or data integrity. Or the impact of the failure harm on the CQA can be neglected. We'll later talk about the CQA. What does that mean? So, or if you can rank it a value of 2, with the impact which has an impact on product quality and data integrity. And the value of 2, what does that mean? Description of that. The failure or harm if affects the product quality, but the consequences can be adequately rectified or compensated in the later process steps to avoid the impact on product quality or data integrity. So if the failure happens, it means that you can still correct the severity in subsequent process steps. So if it happens, you can compensate, you can have some certain mitigation, certain processes that you can apply or certain procedures that you can apply in order to mitigate the impact of the harm. And you can rank it if it has a value, set to rank it a value of 3, then you know that the failure has a critical consequence in the patient safety, then the patient might be impacted, and also the product quality and data integrity. So I usually rank something at 3 if we know that it's going to affect the patient. So the failure harm affects the patient safety, product quality, and data integrity. So all of these 3 together, I consider that as a high severity. So 1, if there's negligible impact. 2, if there's impact on product quality and data integrity. And 3, if the patient also gets impacted with that particular failure, then that is a very high severity. When you rank our description for the currency and probability, so in this case, if you look at if you're manufacturing a product and there's a particular failure that happens, then what's the possibility of that failure? What's the probability of that failure impacting your occurring? So if the probability of the failure is, if you rank it as 1, it means that there's a less than 1 in 50 chances of the failure occurring probability. But if there's another factor over here called uncertainty. That means how well do you know that particular process? Say if you're supposed to mix for 10 minutes and you mix for 5 minutes. Unfortunately, there's a breakdown and the mixing happened to occur 5 minutes instead of 10 minutes. Then that process, how well do you know the impact of that process on your product quality? So if you have done some experiments and you know that if I mix for 5 minutes, it's not going to have any impact on the product quality, the API would have dissolved because the API is very soluble. Then you know, okay, because I understand this process, 5 minutes mixing instead of 10 is not going to affect the answer of my API. So you know the process and you can justify that based on your previous study. Then you can say the uncertainty of this failure happening is, I can rank it as 1 because I know mixing for 5 minutes is not going to impact my product quality, then the uncertainty, you say it's one. So when it comes to occurrence and probability with respect to manufacturing process, there's two things that you need to consider: the uncertainty of the process and the probability. So whichever is high, if the uncertainty value is high, then you say, okay, I need to include this as in place of the probability, because the probability of occurrence is low, the uncertainty is high. So one, if you're looking at the uncertainty or to the process, you can rank one if you have a full knowledge of the process, or you can use two, which is a general knowledge. Basically, you use literature to justify that uncertainty. So that means you haven't evaluated that particular parameter, you haven't done experiments to know the impact of that process on your product quality, but you're using just a general knowledge, which is your theory, what you find in literature, then you can rank it as two. And three, you can rank your uncertainty as three if there's no information, there's no knowledge on the literature, and there is no you haven't done any experiments, and basically you have no idea what's going to happen with that particular uh, uh, failure in the process. So it makes for 5 minutes, you have no idea what will be the consequences. So you rank it as three. But also, if you have an idea what's going to happen, say you have a certain of one, but the probability of occurring of that particular failure is high, say one in every 20 chances, then you're going to use your probability as your ranking. So you're not going to use uncertainty, but you use the probability because it has a high chances of occurring. Then the value of probability is the one that you're going to choose instead of uncertainty to add the quantitative risk assessment. So we look at the severity, and uncertainty, which is uncertainty and probability, which is related to the likelihood of the failure happening. Then I'm going to move to detectability. And this is how quickly can you detect the failure. So the ranking is 1, 2, and 3. I prefer to use 1, 2, and 3 because it's very simple, and the numbers are basically it's just how you define the ranking. 1 is something with a minimal consequence. 2 is something with moderately or medium consequence. And 3 is something with a very high consequence. So that's uh, generally what it is. Some other other um, other approaches, they just like to use 1, 5, and 10. So basically, this is how you define the risk. That's what's most important. And when do you decide to take action? When do you decide, to, okay, I'm just going to take the risk because there's nothing wrong with it. So with the detectability, the ranking is opposite. So 1, that means the failure can easily be detected. So the failure mode can be reliably detected by a direct detection method before or immediately at the onset of failure. So basically, if it's 1, you can say, okay, maybe you can have a visual detection. You can just look at the scenario and say, okay, we expect a failure over there. And then you make the necessary corrections. Or 2 is indirect, where probably you need some instrumentation to be able to detect that failure. So probably in this case, if I refer to the same example of changing material from supplier A to supplier B, and you expect that's going to affect your dissolution. Basically, you won't be able to see a dissolution failure before you use that material. Basically, you need to use the material, make your product, compress the tablet, then send it to the lab, and then you're going to see, oh, the dissolution has changed from 95 maybe to 88. So, which is it requires indirect method, means you need to have an instrument like your dissolution machine to detect that failure. And 3, you can't reliably detect a failure. That means if it happens, it's unlikely that you'll be able to detect it, and the product might just go to the market. So, which means it is hard to detect. So you can see that with detectability, the ranking is the other way around compared to your occurrence and severity. So one, easily detected by directly, before the failure happens. Two, failure can happen, which is you need an instrumentation to detect that particular failure and hopefully be able to correct it so that the product makes the right specification. And three, that means it is likelihood that you're able to detect that failure, and that could have an impact on the product quality or the patient or to the business as well. So that's how we look at detectability. So risk analysis for the quantitative. So it's, like I said, you need to have, you look at the multiple of severity, probability, and detectability. And you can see if you look at the phase matrix, on the left, you see the severity, where there's ranking of 1, 2, and 3, and there's probability with the ranking of 1, 2, and 3 on top, and detectability with the ranking 1, 2, and 3. So if your severity is 1, which is almost negligible to product quality or data integrity or patient safety, and the probability of that failure happening is high, which is a 3, and the detection is very easy, which is with direct method, you can easily detect a failure, then your risk priority, it's going to be 1 for severity times 3 for probability times 1 for detectability. And that gives you a risk priority number of 3, which is your RPN of 3 in the first column. Or if it say, oh, this particular failure has impact to patient safety, so I'm going to rank it at 3 on severity. But the probability of occurrence is maybe or maybe not. There's a medium chance it might occur. So I give it a ranking of 2. And detectability, it requires indirect method. You need to have some sort of instrumentation to be able to detect this failure. So I'm going to give it detectability of 2. So severity 3, probability 2, and detectability is 2. Then it's going to be 3 times 2 times 3, which should be 18, an RPN number. And based on your RPN, you should be able to decide whether this risk is low, medium, or high. So in this case, you can see the RPN of 1 to 3, we've classified it as low, and the RPN of 4 to 9 is a medium, and RPN of 12 and above. The highest RPN you get when you have a severity of 3, probability of 3, and detectability 3. That's 3 times 3 times 3, which comes to 27. And that's your highest. And the lowest you can have is 1 times 1 for severity, 1 for probability, and 1 for detectability, region RPN of 1. So this is how you do your quantitative risk analysis. So you allocate a number, and based on that number, which is a multiple of severity, detectability, and probability, you decide, okay, how you decide whether this is a high risk and requires more attention, medium risk, okay, not so more attention, and low risk, okay, this is depending on the number, and you decide, okay, this doesn't require much attention, we can afford to continue and maybe look at it later. So you can have an example of risk analysis, or quantitative risk analysis. So what is the risk associated with the change of supply of API material from supplier X to supplier Y? So I like to use raw material change because this happens quite often in the manufacturing sector. So if you're making cosmetics, you're making pharmaceuticals, it will come a point where your supplier maybe might be out of stock, and you might need to use another supplier. And this supplier probably have different synthetic processes. The way they manufacture, way supplier A manufactures raw material is different from way supplier B manufacture the raw material. And consequently, these raw materials will differ in their physical chemical properties. So you might need to do your risk assessment. So what we're concerned over here is like, okay, in this raw material B from supplier B, we know the API particle size distribution is slightly different from supplier A. So current settings, which is the specification, not less than 90% is less than 30 microns, and the D50 of the value is 5 to 20 microns. And the bulk density of supplier B, say, let's talk about maybe it's lactose from supplier B, is not more than 0.18 grams per mil, and the loss on drying is not more than 1%. Okay, lactose usually has about 3% moisture, so probably not a good one. Maybe let's use the basic calcium phosphate, which is also a diluent and tend to have less moisture, and it's inorganic. So with regard to particle size, what is the failure mode? So API, okay, we use the API, yeah, not diluent, it's the API. The API has low solubility. So bigger particle size might show a slower dissolution. That's our failure mode. So we change API from supplier A to supplier B, and supplier B has a bigger particle size. And we know inherently the API has low solubility. So we change in particle size from finer raw material for API to a larger particle size API that might affect our dissolution because the API is less soluble. So previous and references, have you done any study to know that the change in particle size might affect your dissolution? No, we haven't done a study, but this is based on general knowledge. So previously or reference, no evaluation study, no. So I just use my general knowledge over here of pharmaceutics and chemistry that lower particle size, larger surface area, bigger particle size, low surface area, so that might affect the dissolution. So affected product quality is the dissolution. So what's the probability of the failure happening if I change from supplier A to supplier B? What's the probability of failure in dissolution to happen? I know changing particle size and the low solubility API will move from finer to coarser, and the API is also will say the probability of the failure happening is high. So that's why I ranked it 3. Okay, what is the severity? I mean, to the patient. Well, is there going to be any harm to the patient? The patient is going to die? No, unlikely, but is it going to affect the product quality? Yes, the product might be impaired because the dissolution is going to change and might be changed to a lower value. And how easily can you detect it? I'm afraid it's not easily detected. I can't see dissolution change before it happens. I'll need to make the product and test the product to understand that, oh, there's a change in dissolution. So the detectability, I'm going to give it a rank of 2. So a multiple of that is 3 times 2, which is 6. 6 times 2 is 12. So risk priority is 12. And based on our matrix, remember, we had a risk priority of 12 up to 27, we classified it as high risk. So I need to have a control plan because it's a risk priority number of 12. In this case, my control plan is track particle size of API and evaluate the impact on dissolution rate. So I'm going to, before I implement the change, I'm going to take this raw material from supplier B. I'm going to do a couple of studies, make a few batches, test the dissolution, and see what's the dissolution look like. Does it really be impacted by a causal particle? Because I know the drug is less soluble, and my assessment was based on just scientific knowledge. So now I'm going to do experiments to actually see, to prove whether, okay, this risk is it really with an RPN of 12? So you have to do some studies to see the impact of particle size on dissolution. If you look at bulk density, so API bulk density shows difference depending on the API supplier. This could indicate the difference in specific surface area. So if something is finer surface area, it has a low bulk density because of by a bigger volume. And if something is mucosa, has a high bulk density because it's by a smaller volume. So we know something with high bulk density might have an impact on your batch size because 1 kilogram of something with a high bulk density will occupy more, high bulk density will occupy less space compared to 1 kilogram something with low bulk density, which occupy high space, a bigger volume. So that might have challenges in your processing. So how did I base it? How did I say that the failure mode will indicate the difference in specific area? So the previous references, the knowledge that I use is production experience show that different bulk densities have no negative impact on dissolution. So it's not going to affect my dissolution, but it might affect my processing. But since uh, it doesn't have an impact on my CQA, which is I'll discuss about my dissolution, which refers to product quality, I'm going to say the probability of failure in dissolution due to bulk density is 2, because I'm just basing it on general knowledge, and the severity is unlikely to have any impact on the product quality, which is your dissolution, so which is negligible, because bulk density dissolution, there's no really correlation over there. And the detectability is of the failure. Obviously, I need to do some lab tests because you need to have an instrument to see if there's a failure in the dissolution. And in this case, I will rank it 4. And in this case, so no further action required. I'm just gonna, test about density as a part of the test before release the product to market. For manufacturing. And loss on drying, that's another parameter of the supplier bleed. The current setting, the current value of the supplier is not more than 1%. What's the possible failure mode? So if the loss on drying has this particular value of not more than 1%, high relative value of drug substance may result in low acid value of the product or the finished product. What do I mean by that? If your API, say, has a purity of, hypothetically speaking, 100%, and your LOD is, say, the LOD is high, is more than 1%, say is 3%. So technically means that the purity of your API will be less because there's 3% moisture, and the dry substance, which is a pure API without moisture, will be 97%. So if you have more moisture in the API, the likelihood of the actual the content of the actual API in the raw material will be less because part of it's been occupied by water. So when you weigh 1 gram or 1 kilo, 100 grams of this API will contain 3 grams of the water moisture, and then 7 grams will be the actual API. So basically, if you weigh 100 grams, you're giving like 7 grams of the API. It's not like 100 grams of the actual API. So that's the meaning of the impact of having high LOD, so which will affect your assay of your finished product. So how do I know this? Well, drug substance is not uh, yeah. So drug substance is not hygroscopic and undergoes wet granulation step. With wetting and drying, no impact on assay is expected. So I know in my manufacturing process, I'm going to use water. And if the drug substance, say, has high LOD, I don't expect it's going to affect the assay because in my process, I'm going to dry the granulation. So that means I'm going to get rid of the moisture. And also, I know that the drug substance as well is not hygroscopic. So if the LOD is different, LOD of the API from supplier B is different from the LOD from of the drug substance from supplier A. So supplier A is 0.5, and supply B is no more than 1%, I'm not going to be too concerned about that particular parameter because I know the API is not hygroscopic and the manufacturing process as well, we're going to dry the granulation. So I'm not expecting any impact on assay. So in this case, the probability of assay being impacted, I'm just going to give it a 1. So it's unlikely that at least this high value of the LOD is going to have an impact on assay of the API. And the severity is, I would say, maybe a 2, because we are yet to test this. I'm yet to use this API, so I don't know what impact will it have on the product quality uh, of the finished product, and how can you detect that the LOD? I mean, how can you detect that assay has been impacted by this particular failure? Then I have to do some tests which require external instrumentation. So it's not like a direct test where you can see with your eye. The detectability requires some sort of instrumentation. So I'm going to rank it at 2. In this case, so your RPN becomes 4. And the control plan is we just continue to monitor that and ensure that LOD of the granulation is within limits. So what I'm going to do, I'm going to use the raw material and make sure that in the drying phase, after using it, I've reduced moisture to the required specification. So basically, this is how you use your quantitative risk assessment to do a risk analysis. So that's basically for now for quality risk management. And what's next is we're going to talk about quality. Now we're going to talk about quality by design. If you recall, I'll say quality by design, where they design a quality product with the respective manufacturing process. And you can do that by building quality into the product by using scientific approaches together with quality risk management. So what are the elements of quality by design? First of all, there's something called quality target product profile. So this is the basis of design of the product. So it looks at it's the qualitative and quantitative characteristics that ensure product quality. So it's the basis of design and development of the product. And you need to consider the following things. So before you design a product, you need to figure out which age group will this be a product be used for? And what's the strength in terms of his API, or if it's a particular cosmetic that has a soothing effect or moisturization of the skin, what's the concentration of that particular material that needs to be in the formulation? And what container am I going to use? Because your container plays a role in your product stability. And all these considerations needs to be based on input from your marketing team. Why do I include marketing team in this aspect? You want to make a product that you're able to sell, that you want people to buy. And the marketing team is the one that's in contact with the market. They know the latest trends. They know where the needs are. They know where the demand is, and they know where the business opportunity is. So they're the ones who have information that can enable you to make a product that can address the market's needs. So your marketing team is very vital in giving an input on what needs to be developed. So that's what I meant by you need to have your marketing team on board when you define this quality target product profile. So they will know, okay, I mean, amongst the children, maybe a certain vitamin is very, very, very popular. And we've seen that it's manufactured by, say, Ricket Bankers. So I think we need to make something similar. And this is their product. So they're the ones who should, give you the feedback, and that should be the basis for you to design the product. And there's also something called critical quality attributes. So these are attributes that need to be within appropriate limits, range, or distribution to ensure product quality. So basically, it's something that ensures product efficacy and safety to the patient. Or if it's a cosmetic, it's something that can ensure uh, product performance, or I know I like to use efficacy in terms of eliciting a pharmacological response, but in the FMCG perspective, probably acceptability, palatability, something that can be it's supposed to do what it's supposed to do, and also it should be safe, and it should have minimal or no undesirable effects. So that's what we call CQA. I did mention this before when I was talking about the quantitative quality risk assessment. And then there's intermediate critical quality attributes. So just like how it says, intermediate, these are intermediate attributes or characteristics that have an effect on the finished product safety and efficacy. We'll get into detail for each and every of these elements and to know, so we can have some relevant example just to give you a deep understanding. And then you have critical material attributes. So raw material and packaging material attributes that need to be within appropriate limits, range, or distribution to ensure product quality. And product quality means by efficacy, safety to the patient. So your raw material, there's certain aspects, elements, properties that contribute to having a product that is safe and efficacious. That means contribute to have critical material attributes, CQA, the second bullet point, that's within limits. That's what I mean by critical material attributes. And critical process parameter. So process parameters whose variation have an impact on the CQA, your critical quality attributes, and therefore should be monitored and controlled to ensure process produces desired product quality. So when you're making a product, of course, there's some process inputs, there's temperature, where there's mixing speed, where there's drying temperature. These parameters may have an impact if they're not within limits, within the appropriate range, which has to be established. I'll later show you how we establish those range. If they're not within the particular range, might affect the product. It might be because they cause maybe deterioration of certain material attributes, and say material, such in this case, it will be critical material attributes. And because deterioration of critical material attributes, that means the material won't be able to give a quality product, the desired quality that it needs to have. That means the product will not be efficacious, or the material might degrade and produce other impurities. It might be of not safe to the patient or consumer. So now there is a big buzz on CBD, cannabis, since it's been legalized in South Africa. I think you've seen a lot of cannabis juice, cannabis vapor, cannabis emulsion. Everything contains cannabis, cannabis, cannabis. Because that's the trend right now. And you expect, okay, probably a marketing team would have noticed this and told you guys, okay, cannabis is a big market, so we need to develop beverages or juice or ointment that contain cannabis oil in it. So he's the one who brings you the information, and probably can bring you a sample product. There's this one which is manufactured by Lot Kingori. It's selling a lot. Let's try to make something similar like this. So that's how you define your quality target product profile. So in this case, probably want to make an emulsion, another emulsion. And the key claim, and this is just an outline of how your quality target product profile can be derived. So your key claim, we want to have cannabidiol oil, which is your active, to be 10 milligram per 5 ml. It should be stable, should be stable for at least 2 years, a shelf life. And we can say that it provides relief to mild to moderate pain, because that's what the innovator or the market leader is marketing it, and how is it used. And the formulation is an oil in water, and composition, it contains eukaryotes, surfactant, preservative, emulsifier, antioxidant, and a dispersion medium. Usually these ingredients will be stated in the container of your reference sample, and the label, obviously, we mentioned the container, how is it packed, and if there's secondary packaging, it will also be apparent in your sample. And that's how you define your quality target product profile. So it's based on what's in the market, based on how you address the market needs. So how do you determine critical quality attributes? I did mention that it's parameters or properties that are responsible, that needs to be within limits, and that are responsible for safety and efficacy of the product. So in order to determine, you need to ask yourself, check if that particular attribute of the product is critical to safety and or efficacy. Safety means that it doesn't cause undesirable effects. That means it should have tolerable side effects or no side effects if possible. And it should be able to do what it's supposed to do. If it claims that it provides relief of mild to moderate pain, it should be able to provide relief of mild to moderate pain. If it says it makes your skin soft, then you should be able to make your skin soft. Then those are what we call critical quality attribute. So if that attribute does not provide any of those, is not responsible for safety and efficacy of the product, then that attribute is not a CQA. So it's not considered as a CQA. And if you say, yes, it is responsible for providing efficacy and safety, then you proceed further and see, okay, can it be altered by process parameters or formulation variables? Okay, by process parameters, what do we mean? Like, okay, if there's a change, say, in temperature, I'm supposed to like we were talking about sucrose earlier, if you're supposed to make it at 95 degrees purified water, I mean, the temperature of 95 degrees for purified water, if I use it at 60, will I still get the same identity of 60 degrees Brie sucrose? If yes, then it's not altered by formulation variable because you change from 95 to 60, you can make a 60 degrees Brie. So then you say, okay, its identity is not altered by formulation variable. But if you change the formulation variable from, I'm talking about process parameter, not formulation variable, process parameter. If it is altered by a process parameter, if you change your process parameter and your quality attribute that attribute is impacted, then you say, okay, then this attribute is critical quality attribute because changing process parameter affect the quality of this attribute. If I've changed from 90 to 60, then the quality attribute also changes and becomes less, it doesn't conform, then you can say then that attribute is a critical quality attribute. If I change, in this case, material, which is your formulation variable, if I change my particle size from 75 to 90, then my dissolution get affected. Then you can say, yeah, that material attribute, that is a formulation variable that can alter that CQA. So basically, to qualify as a CQA, you need to have formulation variables or process parameter variables can impact the quality of that particular attribute. So if yes, you say you identify that as CQA. If unlikely or no, you can still identify the CQA, but you cannot use the risk assessment. So what do I mean by this? I'll give an example, an identity of a raw material. An identity, I mean, say paracetamol. You do an infrared or HPLC to see whether the structure does conform as paracetamol. So that's identity, say IR. Or like, for instance, I think I gave an example of sucrose. 60 degrees Brie. It has to be 60. And if it's not 60 degrees Brie, it will not rotate optical, the optical rotation will not, the degree of optical rotation will not conform to that of 60 degrees Brie. So that's how you identify. So if the process or formulation variable will not, um, okay, let me rephrase. I think this was not the right example, but paracetamol. So you identify by using infrared. So if you change the particle size of the paracetamol from supplier to supplier B, it's only a particle size that has changed. Your identity hasn't changed because particle size does not impact it being paracetamol. It's still paracetamol, but it's a different particle size. If you change, say, your granulation and or you're doing the mixing, and instead of mixing for 5 minutes, you mix for 10 minutes, the identity of paracetamol is not going to change. It's still going to be paracetamol, but the probably the acid might change, but identification is not going to change. So that's what I mean. Something that is unlikely to be altered by the process of formulation variable. It's still considered a CQA because you need it to be paracetamol in order to impart that pharmacological effect, but it's not altered by a formulation variable. So this will not, it's considered CQA, but it's not impacted by processing and formulation variable. So we're not going to be too concerned when we're doing quality by design and when we're evaluating formulation and process variable, which affect your CQ. So that basically that's what it means. So how to define CQA? Does it affect, does it have an impact on efficacy and safety? No, if it doesn't have impact on efficacy and safety, then it's not a CQA. If yes, okay, does process parameter and formulation variable affect these particular attributes? The CQ, yes. If it gets affected by the process and formulation variable, then you identify it as CQA. But if it's not affected by formulation and process variable, it's still considered CQA, but you'll not continue to evaluate in your quality risk management as you're designing your product. So critical quality attributes. So what is a critical quality attribute? We say it's something that's responsible for, needs to be within range, within limits of certain, to be able to elicit, to have efficacy and safety of the product, elicit a certain pharmacological response, do what it's supposed to do, and should have tolerable side effects. So we're going to look at example over here of a critical quality attribute. So you're making, say, syrup, and you want it to have a certain, it has appearance, it has a certain aroma, it has a certain amount of API, probably, okay, let's say suspension, and the particle size has a certain, needs to be a certain amount, and degradation needs to be a certain amount. So these are the quality attributes of your syrup. It's like a description, it involves the color, texture, and aroma. Identification, it needs to be positive for, say, ibuprofen. And your assay, you want ibuprofen to be within 100 plus or minus 5% of a legal claim. So if you have a 2 milligram ibuprofen per 5 mil, you want it to, 100 milligram per 5 mil, you want it to be between 95 and 105. And the part, since it's a suspension, it have a particle size of, say, 90 microns and degradation or impurities within a certain range. So what you need to ask yourself is, does description of my syrup have an impact on efficacy and safety? That's how you decide whether it's critical or not. So we want it to be pink with aroma of strawberry. Okay, pink and aroma of strawberry. Does that have an impact on efficacy and safety? If it's not pink and if it doesn't have aroma of strawberry, does that mean it won't be able to provide mild relief of pain? Does that mean it won't have tolerable side effects? Then the answer is no, because that's description. It has no impact on efficacy and safety. So description, in this case, is not a critical quality attribute. Justification for that criticality that is not critical. So description is not directly linked to safety and efficacy. The target is set to ensure patient acceptability. That's the reason that we have description. This attribute can be effectively controlled by quality management system in the sense that we not identify is critical, but we'll make sure that it does comply because we need patient to be to accept it as to what the one patient complies. Identification, like I mentioned before, we need to be positive for ibuprofen. Is it critical? Yes, it is critical, but it's not impacted by process parameters or formulation variables. Identification, we need positive for ibuprofen because that's the one which has efficacy and assures efficacy and safety of the product. We need to have uh, the correct API. So the justification for saying yes, though identification is critical for safety and efficacy, DCQA can be effectively controlled by the quality management system, and it will be monitored at drug product release. Formulation and process variables do not impact identity. So basically, just need to know after manufacturing, you still have a positive identification for ibuprofen, but we're not gonna be too concerned with formulation variables and process parameters because those we've seen that they do not have an impact on the identification. Assay. Assay is a critical quality attribute. Yes, it is a critical. Why? Because if you don't have the right concentration of your API in your product, you won't be able to get the pharmacological effect. The efficacy won't be there of the product. So we want assay to be within the specified limits. So assay variability will affect safety and efficacy. Process variable may affect assay, and so is formulation variables. Process variable probably if you, in the course of making suspension, you don't agitate enough. You agitate as a mix, you don't mix thoroughly enough for the right time. So you might not have equal distribution of your suspended particles. Or probably if you say mix at the wrong RPMs, I mean inadequate RPM, you might not have the adequate distribution of your suspended particles in your in your container. So assay is a critical quality attribute because it is impacted by your process parameter. Or if your process formulation variables, if your particle size is not of the right size, or you're using xanthan gum, you don't have the right concentration because xanthan gum is supposed to impart viscosity, and suspensions need to be viscous, in order to maintain to reduce the sedimentation rate. So if you don't run the xanthan gum of the right concentration, that could affect your distribution of your API within your system. So yeah, let's say it is a critical quality attribute. Particle size as well. We know that you're going to have the right particle size that could affect your dissolution. So that's your formulation variable that can impact your product efficacy. Degradation product. You want to, each manufacturer of API have a different route of synthetics. So some might use one particular solvent, solvent A, some might use another solvent, solvent B, like your methanol. These are solvents or ethanol commonly used in, or acetic anhydride is a common solvent used in synthesis of API. So if there's a difference in the solvents between two suppliers, you might find that your residual solvents, because you need to evaporate these solvents once you make your API, they need to be within a certain limit. So you may have that your residual solvents are slightly higher from supplier B compared to supplier B. So if it's higher, then it means that in your product, then your products as well, your finished product might have higher residual solvent. Or if you manufacture in your process, you use heat, is temperature. Temperature might facilitate degradation of your API into related substances. These related substances are material that are related to your active ingredient, probably like your starting material. You react with another material to get your API, and when you supply heat, it degrades to your original starting material. Those are related substances, which are also known as part of impurities. It could be harmful to the patient. So you want it to be within limits. So it's either you have high residual solvents, which are impurities which differ from supplier to supplier, or maybe your process parameters might affect the degradation of the API to produce your impurities. So you see that degradation products are affected by formulation variables and your process parameters, and so are also considered as critical. So this needs to be within limits. So all the attributes that have an impact on efficacy and safety are described as CQA. So in this case, degradation product, we say is critical to efficacy. I mean, in this case would be safety, is known as, we consider it as a CQA. Why? Degradation product can impact safety and must be controlled to limit patient exposure. So this is how you have defined the CQA of your product. Okay, so we're going to have a short example over here. Out of all these drug product quality attributes, which ones do you think are critical quality attribute? That means are responsible, have impact on efficacy and safety of your product. Okay, disintegration, disintegration, that means the tablet disintegrates into small particles. Does disintegration have effect on efficacy and safety? I'd say no. Disintegration time has no direct impact on product efficacy and safety, because disintegration is basically a tablet breaking down into smaller tablets. However, dissolution has an impact on patient efficacy, because dissolution is the one that it means the amount of drug that is available into the medium or is available into the body. That means the ability of the dosage form to release the API from the dosage form into the surrounding tissue or the media, and that which can facilitate absorption into the body. So basically, disintegration, no, because disintegration does not entail releasing of the API from the dosage form, but dissolution entails releasing the API from the dosage form and making it available for absorption. So yes, dissolution is critical because it is responsible for product efficacy. Viscosity, no. Viscosity impacts product consistency and texture. That's basically it. It's not responsible for efficacy and safety. Spreadability, I would say in the case of creams and ointment, yes, because you want the product to be in contact with your skin, to stay there. In that case, it will be able to, and diffuse into the localized area to impact its effects. For cosmetics, I think is important. You want it to stay on your skin and diffuse slowly into the surrounding tissue. And of course, for medicated topical preparations, that's where you need to adhere, not run off, and give it ample time to to allow the medication to diffuse into your skin. So yes, spreadability, it depends. It depends with the drug. For topicals, I think it is a critical quality attribute. Content uniformity, yes, content uniformity is their impact on product efficacy, because basically it tells you the amount of the uniformity. It ensures that as the API is in the right quantity throughout each individual unit, and that means that you have the right concentration of API in each individual unit. If you don't have the right concentration, then you won't be able to elicit the desired pharmacological effect, because you'll be underdosing your patient. So that's a critical quality attribute. So material attributes with the potential to have an impact on product CQA. In this case, we mean by critical material attribute. So what are critical material attributes? As we've seen before, raw material and packaging material attributes that need to be within appropriate limits, range, or distribution to ensure product quality. And product quality means efficacy and safety to the patient. So for drug substances, the identity of the drug substance, that's a critical material attribute, because if you don't have the right identity, you won't be able to have the desired pharmacological effect. Potency, the drug substance needs to be as pure as possible. Like for instance, most of the specification, you need to have, it's usually range of 99 to 101 or 95 to 1, no, 98 to 102, or for something which has which has high moisture, like your codeine, is usually 99 on dry basis, 99 to 101 on dry basis, because codeine inherently has about 3% moisture. So as is, it's 97%, but on dry basis, it's 91, 99 to 101. Particle size distribution as well could be critical material attributes, especially for drug API drug substance with poorly soluble, because the particle size determines the dissolution, determines the dissolution. So it also determines how you can undertake the formulation. Probably might need to include surfactant there to facilitate rotability and solubilization into the system. Sterility. If you're making sterile products, like your vaccines, then which are sensitive to heat, the raw material needs to be provided that you're going to use needs to be sterile. So from the supplier, you need to have a supplier that provide sterile material. Or you can have microbial limits, which is applicable to material of a natural origin derived from animal plants. The microbial limit needs to be within a certain limits, so that at the end of the day, your finished products have a microbial limit within the desired range. For excipients, such as preservatives, if you're using preservatives in your formulation like potassium sorbate, methyl paraben, propyl paraben, you need the potency, which is your concentration of your preservative needs to be with the limit. Because if the potency is low, that means you won't be able to provide the necessary preservation efficacy of your product wouldn't have the necessary preservation efficacy, and it will be prone to microbial attack. And your diluent might be your particle size and the particle size distribution. This could be your critical material attributes, because the diluents act as a carrier for small drug substances, or they can also facilitate in the manufacturability, like compaction. When you're compressing tablets, they help to bind the tablets together. So you need to have the right particle size. If you don't have the right particle size, that could affect your compactability of your formulation. Lubricants, like your magnesium stearate, would be interested in the surface area. That could be a critical material attribute, because the way magnesium stearate works, it forms a coat around your particle, around your granulation particles or your powder particles, because it's very, very fine. As you do the mixing, it erodes and starts coating your particle and impart that lubricant properties on your powder particles, and consequently reducing the frictional force, the ejection force between of your tablet. So reduce the friction between your tablet and your dye wall. So the surface area becomes important. So the higher the surface area, the better, but also because that allows the coating of your tablet. So your specific surface area becomes a critical material attribute for your excipient. For your packaging material, you should look at identity, the composition, whether it's glass, PET, or HDPE, because that will affect the quality of the product through interaction. As a primary packaging material, there's a possibility that your packaging material might interact with your finished product, and that could affect maybe a drug concentration, could increase their impurity levels in your finished product, or it could allow maybe your container does allow light and moisture to pass through, and that could affect the concentration of a drug substance if it's prone to oxidation or if it's prone to thermal degradation. I mean, photodegradation. And also if there is migration of packaging material, the composite ingredient, packaging material content into the products. Sometimes some packaging material do interact with the products by releasing its content into the products and hence contaminating your product. Critical process parameter and critical process step. So we mentioned about process parameters, critical process parameters that can have an impact on the CQA, such that when there's a variability in these process parameters, it impacts your critical quality attribute. And this case will be looking at your critical process parameters, which could be your input variables or process step variables, which are these parameters which are associated, which are an indication of the state of that process. Okay, what do I mean? Process input variables, like your drying temperature when you're drying your granulation, your mixing time when you're mixing your powder or when you're mixing your syrup or suspension, your agitation speed. We mentioned that this can have an impact on your concentration or on your loss on drying or on your degradation limits of your finished product. So those are because of an input. And the process step variable, when you're drying your granulation, you want your temperature, say your exhaust temperature, to be within a certain range. Say we want it to be exhaust temperature to be at 30 degrees Celsius. If your exhaust temperature is at 40, that means that your granulation it's an indication that the granulation temperature is too high, because most of the heat is actually not absorbed by your granulation. Your granulation is too high. Most of the heat is being supplied to the granulation goes out. So that means your granulation is too dry, and that could be an indication that the granulation temperature is too high, and could be an indication that your LOD or your granulation might be it's prone to degradation. It will be too dry. So your exhaust temperature is an indirect indication that your LOD might be too low. So it's something that you might need to monitor to ensure that it's within a certain limit, because it's an indirect indication of your LOD values before testing the LOD. And your critical process step. So this is a unit operation that can affect your critical quality attribute. Like for instance, when you're making a syrup, you might need to make up to volume. So you're making 1,000 liters, you've done all your processing and you got to up to 900 liters, and now you're supposed to add enough water to make 1,000 liters. So this process step is considered a pretty critical process step, because if you don't add enough water to make 1,000 liters, say you overshoot to 1,100, that means the concentration of your API in your syrup will be low, because the way the formulation was formulated is to make volume up to 1,000 liters, but you've added up to 1,100. So that means the concentration of the API might be too low. Or if you you're more likely to mostly is over dilution. So that means your concentration of the API is too low, and that will affect your product efficacy. And that's what we mean by critical process step. Like for instance, if you're making vaccines, probably you're preparing a buffer. So buffer preparation step, if there's contamination in the buffer, so it's not sterile. You don't do it under sterile environment, and you end up contaminating your buffer. That's the buffer preparation is a critical process step. And if you don't do it correctly, that means your cells that should be suspended in your buffer will be contaminated and you have microorganism growth, and that will be serious consequence to the sterility and product product microbial limit and consequently patient safety. So basically that's quality by design. Just to have a quick recap, we've seen that we looked at the elements of QBD. We discussed about quality target product profile, look at the CQA, look at the critical material attributes, intermediate critical material attributes, and we discussed about critical process parameter and critical process steps. Now, after this, we're going to look at how we're going to marry the two. We've discussed about the risk assessment and quality risk management. We've talked about QBD. Now we're going to look at how we're going to marry the QBD and the quality risk management, combine these two to develop a pharmaceutical or any formulation and its respective manufacturing process. Right? And it's you'll find some use into it. And if you have any questions or concerns, please don't hesitate to contact Pharmers, and they will reach out to me and I can provide you with the answers. And thank you very much for participating. Well, everyone, that's it. I hope you enjoy the masterclass, and I hope it did have an impact and you'll be able to use what you've learned in your day-to-day developing of formulation and manufacturing process. And thank you very much, and I'll see you next time. Bye-bye.